Good evening, everyone. So I'll just wait for 2 more minutes so that people can join. Then I'll start the session. Meanwhile, can someone comment yes or no whether my voice is clear or not in the chat box? Is my voice clear? Okay, okay. Thank you. So I'll start the session now. Good evening, everyone. I am Pushpangna and I'm a PMRF scholar from IIT Kanpur. So today is the fifth week of the problem solving session for the NPTEL course Cell Culture Technologies, which is being taken by Professor Manakas. So I'll just introduce you to some basic model questions, so which are going to help you in solving this week's assignment, the week 5 assignment. So this is the first question. So papain is an enzyme which is derived from, and we also have to mention what kind of enzyme it is. So is it a proteolytic enzyme, or is it a lactose dehydrogenase, and is it derived from papaya or from milk? So yeah, papain is a proteolytic enzyme and it is derived from papaya. So the structure that you are seeing here on this side, this is the structure of like the basic domain architecture of papain. So whenever we visualize any protein in this kind of an arrangement, where we can see the distribution of amino acid, like here you can see from amino acid 20 to 340. Before the amino acid 1 to 20 is just the signal peptide. So this kind of arrangement of any protein is called domain architecture. So this is the domain architecture of our proteolytic enzyme, which is papain. Now, another important thing to know about papain is that it is a cysteine protease, and as you can see in the domain architecture itself, that it has these disulfide bonds, which are due to presence of cysteine, and this is what the basic distribution of the protein or the enzyme looks like. Here we have the activation peptide and then we have the activation site. Now, what is the purpose of papain? It is a protease. It is basically an endopeptidase. So, as the name suggests, endo means it's not going to cut at the ends. Rather, it is going to cut in between. So here, as you can see, for this antibody, so in the antibody we have this Fc fragment and the Fab fragment, right? So here, papain acts between like this Fc and this Fab region, and it cleaves in between. So sometimes in some experiments, in some biological experiments, we do not need these Fc fragments, because Fab fragments are those fragments using which most of the experiments are carried out. So for that purpose, this enzyme papain is used. And you must have heard about another enzyme, which is pepsin. So pepsin cleavage is different. So for different purposes, we have these two different kinds of enzyme. But the enzyme in question here, which is papain, is used for separating the Fab fragment from the Fc fragment. Now next question. Which of these protein misfolding and aggregation is linked to Alzheimer's disease? So is it amyloid beta? Is it tau protein? Is it both A and B, or none of the above? Yeah, so Alzheimer's disease is caused by misfolding and aggregation of both amyloid beta and tau protein. So the correct option is both A and B. So Alzheimer's is basically a neurodegenerative disorder, and it usually results in memory loss or memory impairment. If you see the comparison between a healthy brain and an Alzheimer's brain, here you can see this is the healthy neuron, and but on the right side you have these protein aggregates present. So aggregation of these proteins, or amyloids as we call them, are a major cause of Alzheimer's. So here again we have a comparison. You can see that the neurons are normal here, like for a normal brain. However, for a patient suffering from Alzheimer's, you can see that there are these amyloid plaques which are present between the neuron, and also there are these tangles which are present within the neuron. So these are formed by amyloid formation and also by aggregation and misfolding of tau protein. So the next question is, in a hemocytometer, volume of 1 millimeter into 1 millimeter square is, first is 0.25 nanoliter, second is 5 nanoliter, third is 60 nanoliter, and fourth is 100 nanoliter. So I'll tell you the answer first, and then I'll explain why that answer is correct. So here the correct answer is D, which is 100 nanoliter. So if you look, so this is a hemocytometer or a Nobel chamber, as many people call it scientifically. So this is the grid where you sample, place your sample or your solution, and then suppose you are putting 10 microliters or 20 microliters of your solution here. So here your solution will be covered, like your solution will cover the space. If you zoom in to these two chambers, then you see that each chamber is made of something like these kind of squares. Like if you look under the microscope, then you'll see that it has these kind of squares. Now here, so there are basically 9 large squares. So this is one. So these are the 9 large squares, and each square is 1 mm into 1 mm. Suppose we are considering this square, then each square is 1 mm into 1 mm. Now, uh, your question is, what is the volume of this large square, which is 1 mm into 1 mm? So for knowing the volume, we also need to know what is the height. So like, so basically the height, like the distance from the cover slip to till your chamber here, so we have this cover slip here, is approximately 0.1 mm. So this is how hemocytometers are made. This is predetermined, that it is 0.1 mm. So now you know that length and width, which is 1 mm, 1 mm, and you also know the depth or the height, which is 0.1 mm. So from here you can calculate the volume. So basically then it will be 1 into 1 into zero point 0.1 millimeter cube, right? If you convert it to centimeter cube, then it will be somewhere around 10 to the power minus 4 centimeter cube, right? Now 1 centimeter cube volume is approximately 1 ml. Okay, so here that means here we have 10 to the power minus 4 centimeter cube, which means we have 10 to the power minus 4 ml. So this will be what? Here it will be 100 nanoliter. So each of this large square has a volume of approximately 100 nanoliter. So I'll show you a demonstration of like how actually we do cell counting with hemocytometer, and after that I'll give you a numerical. So please pay attention to that numerical, because there is also one question in your assignment that will help you to solve it. First, like let's watch this demonstration, and after that we can solve that numerical. So here, as you can see, this is the hemocytometer, and here from the pipette, you can see the technician adding the solution, and your cover slip is here. So this is your cover slip, and this is the pipette through which, like this is the area through which solution is being added. Now what happens? A widely used stain for visualizing cells under the microscope is the trypan blue stain. So the blue color that you are seeing here, which is coming in the solution, is because of trypan blue. What trypan blue does is, it's going to stain dead cells. So your dead cells are going to appear blue, but your live or your viable cells are going to appear as they are, as it is. And that is because trypan blue can percolate through the membrane of the dead cells, but it cannot go through the membrane of cells which are viable or which are living. So whenever under the microscope, if you are going to see blue cells, that means those cells are dead, but the cells which are not blue, those are viable. So in order to determine the viability of the cell culture, we use this trypan blue stain. So basically what is the routine procedure which is followed in labs is, the culture, whatever the volume of culture you are taking, suppose if you are taking 100 microliter of your cell solution, then to that you add 100 microliter of trypan blue stain. So from here, that means what will be the dilution of your cell. So it will be like the total amount is 200, like 100 plus 100, and but in that only 100 microliter of your cells is present. So we do this 1:1 dilution. That is routinely done, but different labs have different procedure. So I'll start the demonstration now. So you can see how it's going in the chamber. We usually add around 10 microliter solution in the hemocytometer chamber. So you can see in the Eppendorf, we already have the cell solution, and to that we are adding say 100 microliter of trypan blue. And once it's properly mixed, 10 microliter is taken and added to one chamber, and similarly another 10 microliter can be added to the other chamber. So you can see there are two chambers here. Now if you look under the microscope, so here this is how a hemocytometer grid looks like. As I told you earlier, there are 9 large squares. Now since we added 10 microliter solution in one chamber, that means we have 10 to the power minus 4 ml of solution. So for counting, what we do, we use the like 4 corner squares plus 1 center square. So here this square, this one, this, and this. Now here you can see that we are counting viable and non-viable cells as well. So as I told you, viable cells are going to be those cells which are not stained blue, but your non-viable cells are going to be the ones which are stained blue, which is this one in this case. So here we have 10 viable cells and 1 non-viable cells, but this is the case only for this square which is outlined in yellow. We still have to count 4 more squares. So it's a different practice in different labs. Some labs just prefer counting just these 4 corner squares, and some labs prefer to count 4 corner squares but also the middle square. So now we have moved on to counting in the second uh, top right corner square, and then the bottom right. So it's usually you have to move in this kind of zigzag pattern, like this Z pattern, so that you do not lose count of from where you are counting. And people usually have a cell counter, which is a normal counter, because just remembering what is the number of cells is going to be difficult. So now we have this center square. So these are two cells which are clumped together, but you can clearly see that those are two cells. So we are going to count those cells as two rather than just one, because it's clear that those are two. So it was counted as two cells. So now our counting is done. So the total viable cells that were counted were 54, and the non-viable cells, the ones which were stained blue, were 3. So now if I ask you to calculate the percentage of viable cells, that you can easily do, right? Because number of viable cells 54, the total number of cells will be 54 plus 3, because total will be everything, living plus dead cells, both kind of cells. So now the percentage of cell viability is around 94.7%. Second thing is to count what is the average number of cells per square. So we counted 5 squares. Had we counted only 4 squares, it would have been 54 divided by 5. We are not taking 57 here because we only have to count the viable cells. So per square, we have around 10.8 cells. Then we have to calculate the dilution factor. So your dilution factor is going to be your final volume, which is 200 microliter divided by 100 microliter, which is the volume of your cells. So here the dilution factor is going to be 2. Now your concentration of viable cells per ml in your culture, which is the most important point to determine, that will be your average number of cells per square into dilution factor into 10 to the power 4. 10 to the power 4 because that is the volume that we added. So here it is this number of cells. If we are going to write it in scientific notation, that is 2.16 into 10 to the power 5 cells per ml. So this is your viable cell count. So hemocytometer is used for determining the viable cell count. It is one of the most important and basic techniques in cell culture that you will have to use in any kind of cell culture lab you go, no matter what kind of study you are doing, and that is to determine the viable cell count using a hemocytometer. So it's a very important concept and a very important topic, and you should know how to determine that, because without that you will not you will not be able to determine what is the specific concentration of cells that you are going to seed, what is the concentration of the cell culture that you are beginning with. So that is why it's important to determine this. So I hope this demo was clear. Now I have a numerical. We have this kind of numerical in your this week's assignment as well. So we can solve this. So the question is, you take 100 microliter of cell suspension and mix it with 100 microliter of Trypan blue. So we have 100 microliter of cell suspension and 100 microliter of Trypan blue, same as in the previous demo. Of this, you load 10 microliter into the hemocytometer. 10 microliter means in one chamber of the hemocytometer, you load 10 microliter. And when you do the counting, you get 400 cells. But in this question, we are counting only the 4 corner squares. We are not counting the 5 squares, only 4 corner squares. So the total number of cell is 400, of which it is saying that 40 are dead cells. So that means what is our what is the number of our viable cells? that we can determine by total number of cells minus the dead cells. So what is the viable cell count per ml? So I'll wait for 2 minutes. You can try solving the question and write the answer in the chat box, and then I'll explain how it is being solved. So I'll just wait for a minute or two. So you have to determine the viable cell count per ml. This 400 minus 40, 360 will just be the total number of viable cells in the 4 squares. But that is not going to tell you what is the viable cell count per ml. So you have to determine this. For doing this, you have to calculate the dilution factor, the average number of viable cells per square, and after that, at the end, you will calculate the viable cell count per ml. So basically here, the what is the number of viable cells? So the number of viable cells will be total cells we have 400. Out of that dead cells are 40. So our viable cells are 360. Now we have 360 cells. So in the question, they have only taken these 4 coordinate squares. So we have 360 viable cells in 4 squares. So first of all, what we can do is we can determine the percentage of viable cells. So this will be easy. This will be number of viable cells divided by total number of cells into 100. So this will be 90%, right? So this is your percentage of viable cells. Then you have to determine the average number of cells, that is average number of viable cells per square. So from here you can see we have 360 viable cells in 4 squares. So per square means in 1 square we we have approximately 360 by 4, that is 90 cells. So on an average we have 90 cells per square. Now what is our dilution factor? So as I showed earlier, dilution factor is going to be the total volume of like the solution divided by the volume of cells. So here the dilution factor is 2. So if now we have all the information, we have the dilution factor, we have the average number of cells per square. So now what is the cell count or cells per ml? So if we put it in the formula, average number of cells, n here is the average number of cells per square into the dilution factor into 10 to the power 4 per ml. This 10 to the power 4 has come from the fact that we are adding 10 microliter, we are loading 10 microliter in the hemocytometer. So it has come from there. So what will we have? The average number of cells per square is 90, dilution factor is 2, and 10 to the power 4 per ml. So we have around 1.8 into 10 to the power 6 per ml. So this is going to be your answer. Someone already calculated. I can see in the chat box. Yeah. So this is your viable cell count. So you have a question like this in your this week's assignment as well. I hope you can solve it now, or if you have any doubt, if you had any doubts, those are clear. So moving to the next question, which of these antibodies is used to label mostly neuronal cells? So is it MHC, MAP2, islet, or anti-RYR? Should we need to add media added to cell suspension to calculate? No, we do not need to add media in order to calculate. So basically, the cell suspension that you will be taking will have some amount of media, right? Because like it will be growing in media itself. So that is why we are taking, like, it doesn't matter in which medium or where your cells are. You just have to determine the volume. So suppose if we are taking 100 microliter of cells, that means 100 microliter of media which has cells. Okay, because it's in a solution form. We do not need to add any media from outside, just like we are adding trypan glue, because cells are already in media. So I hope your doubt is clear, Dr. Deepa Armani. So for this question, which of these antibodies is used to label mostly neuronal cells? That is MAP2. So sorry. So MAP2 is an antibody which MAP2 is a mature neuronal marker. So it's a widely used and very common neuronal marker for mature neuronal cells, and it is a neuron specific cytoskeleton protein. So the correct answer is MAP2. Now here, suppose I do immunohistochemistry or antibody staining of my neuronal cells with MAP2 antibody, then this is what, like, this panel that you're seeing on the left, this is what it's going to look like. You can see the neurons here, and these blue dots that you're seeing is for the MAP2 antibody, which is a fluorescent antibody, and you can see clearly that it's marking the neurons. Next, if you are going to do Western, so this is for immunostaining. If you are going to take your cells and run a Western, for example, you can see from here mouse brain and rat brain, and then if you use MAP2 antibody, then these are the kinds of bands that you are going to see. Next, if you are going to do a immunohistochemistry study, for example, here in a rat brain or rat cerebellum, then this is like these dots that you are seeing. This is a staining protocol. The brain tissue has been stained using the MAP2 antibody. So this is how you are this is how it's going to look like under the microscope. So for seeing neuronal cells, this MAP2 antibody is used. So the next question is, neurobasal medium is buffered by basic amino acids, oxygen, sodium bicarbonate, or glucose. So, neurobasal medium, as the name suggests, is used for growing or culturing neuronal cells, and it is buffered by sodium bicarbonate. So, this is the basic composition of the neurobasal medium. It has these essential amino acids along with some non-essential amino acids and other components such as glucose, hepace, phenol red, which is an indicator, sodium pyruvate, then vitamins, and then in the inorganic salts, you can see that it has sodium bicarbonate. So your neurobasal medium is actually a medium which uses the CO2 and bicarbonate buffering system, which I discussed in the previous lectures. And so this is an image which shows neuronal cells which have been grown using a neurobasal medium. So in the very first week, you can see there are not as many cells as there are in 3 weeks, which have formed those extensions. So moving to the next question. So xeno-free medium. Xeno-free medium. So there are 4 statements about this xeno-free medium. So xeno means basically something which is coming from outside, right? Something which is, so for our cell culture needs, xeno means something which is coming from animals, which is not human. So a xeno-free medium A does not contain ingredients derived from non-human animals. B does not contain recombinant materials which are made from non-human animal DNA sequences. C may contain purified, processed or unprocessed materials from human sources. D may contain recombinant materials made from human, plant, bacterial or yeast DNA sequences. And E, animal-derived components may have been used as raw materials at the secondary or tertiary level of manufacturing unless otherwise indicated. So then you have to tell which one of these statements is true and which one is false. So again, as the name suggests, xeno-free medium, as I explained, will not have any ingredients which are derived from non-human animals, basically which are derived from animals. So this statement is correct, right? So a xeno-free medium will have ingredients, will have serum and other stuff which are derived from humans only, not from any other animals. Like for example, in cell culture, the most commonly used serum is our bovine serum, right? Bovine serum or horse serum. So these are the commonly used sera in cell culture media. For example, in DMEM, we add 10% serum. We use this FBS. This is the term which you will hear routinely in cell culture labs. So FBS is fetal bovine serum. But if we add FBS to our system, to our media, then it will not be a xeno-free medium because it is this serum is derived from bovine. So this statement is correct. A xeno-free medium will not contain ingredients which are derived from non-human animals. The next statement does not contain recombinant materials which are made from non-human animal DNA sequences. This is also correct because it is xeno-free. It will not have ingredients which are derived from non-human animals, and that means it's not going to have recombinant materials from animals as well. Then third option, may contain purified, processed or unprocessed materials from human sources. This is correct. It's xeno-free, but we want to have, so basically we use xeno-free medium in laboratories, that is because we want to do a translational research where we can move from where like whatever we are doing in laboratory can directly move from laboratory to clinical trials. But for clinical, suppose you are growing and you are testing everything in a small scale laboratory, but there you have these systems. You have fetal bovine serum, you have horse serum, and you have some other ingredients which are derived from animals. But when you're going to go for clinical trials, there you cannot supply fetal bovine serum for treatment for to humans, right? So there you need to switch from like these animal based sources to a source which uses ingredients which are compatible with humans. So, and that is why when most of the laboratory results go to clinical trials, they fail there, because in a laboratory you are using ingredients which are different from what can be used in a real world scenario. So that is why people like want to shift to using xeno-free mediums nowadays. So yes, this statement is also correct. It can have materials from human sources. Then the D option may contain recombinant materials made from human, plant, bacterial or yeast DNA sequences. This is also correct. And then fifth option, animal derived components may have been used as raw materials at the secondary or tertiary level of manufacturing unless otherwise indicated. So this is also correct. Animal derived components may be used somewhere as raw materials, but not at the primary level, but at tertiary or secondary level of manufacturing unless indicated. So here for xeno-free medium, all these statements are correct. From A to E, all are correct. So moving to next question. Leibovitz L15 medium contains what and lacks what? So basically this Leibovitz L15 mediums contains sodium pyruvate and it lacks sodium bicarbonate. So it is a CO2 independent medium, and that is because for its buffering it does not need CO2, and that is why it does not have sodium bicarbonate. It can buffer using normal basic amino acids and sodium pyruvate. Now moving to the next question, which is which of these brain regions is associated with memory and learning? So memory and learning are associated with the hippocampus. So very briefly, I'll move on to the parts of the brain. I'm sure like everyone has read this, but very briefly I'll cover just for a recap. So this wrinkle part, this blue wrinkle part which you are seeing is the cerebral cortex. So this cerebral cortex is the largest part of the brain, right? So here on the right side, you can see like for each part, it's written what kind of function it is involved with. So a part of your thinking, movement, touch is determined by the cerebral cortex. Then at the back of the brain, we have this cerebellum. So cerebellum is responsible for movement coordination. Okay, so that you make smooth coordinated movements like walking, running, writing, or things like that. So it is responsible for proper movement, the cerebellum. Then here we have thalamus. Okay, so what thalamus does is that thalamus is responsible for your senses and for your consciousness, right? So all your sensory things which are happening, like smell, hearing, those things are taken over by the thalamus. Then we have the hippocampus. Now, hippocampus, which is here, hippocampus and the cerebral cortex, they work together to make your memories and your thinking so that you remember everything. So here, as you can see, memory processing is done by hippocampus. Then next here we have the hypothalamus, which is here. So again, your hypothalamus will govern emotions, your circadian rhythm, and again your memories. Then here this pink part which you are seeing is the corpus callosum. So corpus callosum basically connects the right hemisphere of your brain to the left hemisphere. You know that, right? And the cerebellum also is right and left. So the only job of corpus callosum is to connect the two hemispheres of the brain. Then we have pituitary gland, which is very tiny. This red part which you are seeing. So pituitary gland is the smallest part of the brain. The biggest part is the cerebral cortex. Pituitary gland is the smallest. And what pituitary does is it controls your hormones. Okay. Then we have the brainstem where we have midbrain, pons, and medulla. So all these perform different functions, but basically they perform our heart rate, breathing, blood pressure. Then from here we have the spinal cord. This is complete. Here we have the spinal cord, and so what spinal cord does is it connects your brain to the rest of your body, and it is through spinal cord that most of your messages pass from your brain to the rest of your body. Okay. So moving to the next question, what is the characteristic shape of hippocampal neurons? So there are four options. Is it round, pyramidal, elongated, or hexagonal? So hippocampus, as I mentioned earlier, is involved in memory. So hippocampal neurons are characteristically pyramidal in shape. So you can look at the diagram here. So this is a microscopic image of the hippocampal neurons. And here the hippocampal neurons are expressing YFP or yellow fluorescent protein. That is why you are seeing everything yellow in color. So you can see the basic pyramidal shape of the neuron, and this is a zoomed in microscopic view of just one hippocampal neuron. Here there are many. So basically, hippocampus works on forming new memories, and these hippocampal neurons play a very important role in forming of those memories and also detection of new surroundings and stimuli. So again, I'll show you a video where it's a very basic video of how hippocampal neurons are cultured, like how the brain neurons are cultured. So this is your brain. Then here you have your hippocampus, both in the right and the left part. And then you extract those cells. You make single cell suspensions. So you get the cells in a tube, then you can count using hemocytometer, like what is the number of your cells. Then you can plate those cells in your petri dish, use specific media such as the neurobasal media that we discussed, and then so this is the basic flow. So now next I'll show you, like suppose you have grown your you have plated your neurons in your petri dish. Now how does it grow? Like you made single cell suspensions, right? So you have individual neurons. You So how are they growing? So this is a time lapse video where they recorded the neurons growing after they had plated the neurons. So you can see how it's growing. Like it's forming those dendrites, is forming the extensions. So the black part, you can see that those extensions are growing, they are extending. So this is phase contrast microscopy. Now you see it's growing. So basically when you plate neuronal cells on your petri dish, after some time, if you look in the microscope, you see that they have grown. So basically this is how they grow. You can see it was so small, but now it has extended, that it's going out of the frame. And once you give it proper time to grow, it will cover your entire surface of the petri dish. So the next question is, which of these is not a developmental stage in rats? The first is embryonic, prenatal, rental, or neonatal. So if you look at the developmental stages of rat or a lab rat, so this is the prenatal developmental stage, which again is divided into germinal, embryonic, and fetal stage. And once the fetal stage is over, then comes the postnatal stage. Like you have the neonatal stage, then the weanling, then peri-adolescent, then you get the young adult. So these are different stages. However, there is no such stage as this rental stage. So the correct option is rental stage is not a developmental stage in rats. So this was all for the questions today. Uh, all the videos and all the sessions that I'm taking and uploading on my YouTube channel. I'll again post the channel link on the chat box. Just a minute. And from here you can access the recordings of the videos. So I posted the link of the playlist which has these recorded videos on in the chat box. If you have any questions which are related to the questions which I discussed right now, you can put it in the chat box. And if this meeting doesn't end, then I'll try to answer those questions. Presence of which of the following will disqualify a culture medium as defined? So if I'm talking about something which is defined, so define means you know the exact number or the exact percentage of anything which is going to be added in your medium. So your options are known quantities of growth factors that is going to be defined because the quantity is going to be known. And then second option is phosphorus buffered saline. Again, you know what is being added and you are going to know what is the amount that you are going to add. Then serum and vitamin A. For vitamin A also, you know that it's vitamin A which you are adding. However, in a serum, you do not know, you do not know what exactly is the amino acid composition and what exactly is the composition of the serum which you are adding. So, if you add a serum, then your culture medium will cease to be defined. So, the correct answer is C, serum. Okay. In the assignment question, we got the link from picture of hemocytometer in that we could see some cells and crosses. Do we need to consider those cross as non-viable cells? Because number of viable and dead cells were not mentioned in the question. Okay, so I do not know what the cross means. Okay, if in the assignment it's not mentioned that what is the like if it's not mentioned that you have to consider viable or dead cells, then you cannot consider it. But since I think in that question it mentions that we add trypsin blue, so so basically, I think in the assignment grid which is given for the hemocytometer, that is just to show your hemocytometer. The specific number of cells which are there is mentioned, right? So you do not need to consider viable or dead cells. You can just think that the number of cells which are mentioned in the assignment question is viable, and you can like calculate accordingly. I'll see if I can open that uh, figure, that hemocytometer figure from the assignment. The neurons of which part of brain are mainly affected in Alzheimer's disease? I think it's basically the hippocampal region because that is from where it starts. But subsequently, I think it starts affecting the entire nervous system of the brain. So I'm not able to open that figure here, but for the chemocytometer question. But since like dead and viable cells are not mentioned, you consider the number which is given in the question as the viable cell count and you calculate accordingly. Can you upload today's session? Yeah, I'll upload the session today on that YouTube link which I have like added in the chat box. So this is the uh, YouTube channel where it will be uploaded. So thank you all for today's session. I hope it was informative for you. Next week also there's another session which you can join.